Okay, nobody wants to listen to me chatting. You want to hear from our amazing speaker. I will introduce her in just a second. But you can see that her talk title is Leveraging Brain-Based Predictive Modeling to Study Sex, Gender, and Behavior. And believe me, I've heard her speak and she's amazing. So you're totally in for a treat. Before I begin, though, or either one of us begins, I want to make a land acknowledgement. I want to recognize that many of us live, work, play, and participate in a community on the unceded, ceded, and traditional territories of the 630 First Nations, each of which possess their own unique traditions and history here on this land that we now refer to as Canada. I also recognize that many of you are coming from other region. So I hope that you look up the Indigenous peoples of the lands on which you now dwell. Here in Canada, we want to acknowledge the importance of the Truth and Reconciliation Commission of Canada's call to action and the United Nations Declaration on the Rights of Indigenous Peoples Act. In all of our work, we are committed to ensuring Indigenous women's rights to health and safety and the equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. Today, I am joining you from Toronto, which is covered by Toronto Purchase Treaty Number 13 with the Mississaugas of the Credit. Toronto is now home to a vast diversity of First Nations, Inuit, and Métis who enrich the city. And many of our members and many of you joining today are likely in Vancouver, which is part of the traditional unceded homelands of the Coast Salish peoples, the Musqueam, Squamish, and Tsleil-Waututh First Nations. This is brought to you by the Women's Health Cluster. We have over 470 members, including faculty, trainees, and community members across 26 countries worldwide, which is really amazing considering we just started this in 2019. So it just goes to show you that we need organizations such as this. We have a number of events. Please check out our website, which is down below. And especially for trainees, we have support for traveling to conferences. I think the next one will be in November, the next announcement, and we have very good success rates, so please do apply. You need to be a member to apply. It doesn't cost you anything to become a member. Please reach out and look at those opportunities. And we also have podcasts and blogs that I also encourage you to take a look at and make yourselves available of the things that we offer, including we have a YouTube channel. So I've stopped sharing, so Elvisha can start sharing her screen. You might want to go off mute first, as I have discovered, while I introduce her. I don't know if that works for you. You're more computer savvy than I am. Are you disabled? Or you can, in terms of, can you host?
Yes, it was working earlier and I don't know what it's doing now, but we'll figure it out.
That's just nice. Well, that could be because, it switched to me as host, but you are co-host, so you should be fine. So while Elvisha, I never asked how to say your last name. So Dhamala? Dhamala. Is a PhD and assistant professor in the Institute of Behavioral Sciences at the Feinstein Institutes for Medical Research and the Zucker Hillside Hospital at Northwell Health. And if that's not enough, the director of the Brain-Based Predictive Modeling Laboratory. Her research seeks to characterize the neurobiological correlates of human behavior with an emphasis on understanding the extent to which these brain behavior relationships are shared or unique across males and females in healthy and psychiatric populations. This research focus is largely motivated for the search for gender and sex-specific neural underpinnings of complex behaviors, which may underlie distinct manifestations of affective and psychotic psychopathology in males and females. And as I said, Elvisha, you are on your presenter mode, I think, so you probably want to swap screens.
Oh. If I can figure out how to do that.
Yes, sometimes it's at the yeah, you did it. That's great. And Elvisha, it's not on your bio here, but you, I think, obtained you're Canadian. This is what I know, although you're living in the US right now, and you got your PhD from McGill or undergrad?
Cornell.
Oh, Cornell.
I did my undergrad at
McGill, Cornell, whatever. One of the top institutions in the world. So Elvisha, I know, is Canadian, also because her bio has a bunch of Canadian spellings of behavior. So that also was great to see. So I always ask for fun facts. And Dr. Dhamala's fun fact is that she's lived in 10 different cities. So I guess maybe I could be forgiven for thinking that maybe you got your PhD. Who knew? Who knows where you got it? And what are those 10 cities, if you don't mind telling us?
So I have some of them actually in my career overview, so you'll get to
Oh,
hear
you'll
a bit
get
of
it.
them.
Okay, great. Well, we look forward to hearing you talk. Thanks so much, Elvisha, for agreeing. Take it away.
Amazing. Thank you so much for the invitation, and thank you everyone for being here today. So today, as Lisa mentioned, I'm going to be talking about how we can leverage brain-based predictive modeling to study sex, gender, and behavior. So if I can, okay, there. So I'll start off with a brief overview of me, how I got here, and then talk about predictive modeling, neuroimaging, and specific studies where we've used predictive modeling and neuroimaging to study different brain behavior relationships pertaining to different behaviors, as well as gender. And then I'll end off by talking about the implications of this work. So, as a brief overview, I was actually born in Nepal and I moved between there and the US early on. So that kind of covers like 3 different cities. And then eventually I moved to Toronto when I was 8, which is where I mostly grew up. And like I said, that's part of the reason why I've lived in so many different places. Another fun fact about me is that I was obsessed with Shrek growing up. And this photo was taken the same year the original Shrek movie was released. And I was showing these slides actually to my partner this weekend, and he told me how creepy Shrek looks here. But I like Shrek, so I'm gonna leave it there. And a lot of these early experiences, kind of moving between different countries, different cultures, helped me realize that kind of no matter where you went in the world, there are always these underlying ideas or preconceptions of what males and females should be and how they should behave. So that definitely shaped a lot of my research interests, even before I knew I was actually interested in research. So moving on to the next stage, I did my undergrad at McGill and I majored in neuroscience. And at the time, I thought that I would go to med school because that's kind of all I thought you could do with that. And I started working in different research labs. I worked in a electrophysiology lab where I took care of like weakly electric fish. I did immunohistochemistry, looking at chick embryo brains. I did MRIs on people to look at the different correlates of memory. And kind of doing all this, I realized that I didn't like any of it. Mostly because I realized I did not want to work with animals, did not want to work with cells, did not want to work with people. So then after I finished undergrad, I spent 6 months working as a research assistant lab. And there I got to just analyze imaging data without having to actually collect any of it. So I was like, this is great. I can work from my computer. I can work for wherever. So then for my PhD, I did it in neuroscience at Weill Cornell Medicine, which is in New York City, but I spent time between there and Cornell's campus in Ithaca. So going into grad school, I thought I wanted to go into industry, and to kind of open up my options, I was like, I'm going to do machine learning because that's what all the cool people are doing. My thesis specifically focused on machine learning and brain-based predictive modeling, which I'll be talking about today. But then I graduated during COVID and no one was hiring anyone, especially if you were international. So I figured like I could do a postdoc. I'll see how this goes. So then I went to Yale for about 1.5 years to build upon the prior research that I'd done and expand it to consider more clinical behaviors. So specifically expanding it to look at psychiatric illnesses, which have a lot of these sex differences that I was interested in. About 1 year in, I knew I didn't want to be a postdoc forever and my lab was going to be moving to another institute. But again, it was still COVID and no one was hiring. So I figured I'd apply for faculty positions and see how it turned out and then consider industry as an alternative. So that brings me to now, when I started my lab here about 7-8 months ago. And now my lab's research is primarily focused on using brain-based predictive modeling to study sex and gender differences in brain behavior relationships. So I hope that gives a bit of context as to who I am and how I got here. So now I'll get into the research side of things. So white matter fibers in the brain directly and indirectly connect different brain regions. These structural connections facilitate the flow of functional activation patterns between different regions. And together, these functional and structural relationships have primarily been studied at a group level to understand group differences between healthy and clinical populations, males and females, children and adults, and so on. But with increased access to big data and computational resources, we can actually now use what we call predictive modeling approaches to understand how neurobiology underlies psychopathology and behavior at an individual level. Specifically, we can use data to learn these relationships at a group level, and then use that to perform descriptive, predictive, and prescriptive analyses to get individualized predictions about illnesses and behaviors. Descriptive analyses rely on existing observations to identify underlying patterns to describe and summarize the data in an easily interpretable manner. So these machine learning approaches can be used to reveal broad trends in neurobiological, behavioral, and clinical phenotypes, and have been used to identify data-driven correlates of illnesses and behaviors and data-driven subtypes of illnesses. Next, we have predictive analyses, which establish associations between independent and dependent variables and use those learned associations to predict trends and properties in unobserved samples. So we can use these to identify future risk for the development or deterioration of illness in a personalized manner. And we can use these to predict clinical, cognitive, and personality traits across different psychiatric illnesses. Finally, we have prescriptive analyses, which enable us to recognize risks and offer potential treatment solutions. These approaches identify anomalies within the brain and suggest personalized interventions that can most effectively alleviate individual-specific patterns of concern. And these can be used to predict clinical responses to medications, as well as responses to cognitive behavioral therapy across different populations. So, for example, you could use someone's brain connectivity patterns to predict how likely they are to respond to a given medication. And together, these descriptive, predictive, and prescriptive analyses can be used to get clinically meaningful insights that can then facilitate the development of personalized diagnostic tools and therapeutic interventions. In my research, I'm primarily interested in using neuroimaging data for predictive analyses. So basically, I'm using information about the brain to then predict individual differences in diagnoses, cognitive abilities, personality traits, and dimensional symptom profiles. So I'm going to shift gears a bit and talk about neuroimaging, and then I'll come back to this idea of predictive modeling. So neuroimaging is a technique that's used to directly or indirectly image the structure or function of the brain. There are a lot of different techniques out there for in vivo human brain imaging, and some of the ones are, and they're shown here, are based on their spatial and temporal resolutions. So there are some techniques with higher spatial resolution used for local brain imaging, whereas others work for whole brain imaging, but at a lower spatial resolution. They also vary in terms of the time scale or the temporal resolution. And in my case, I'm interested in the whole brain correlates of behaviors, so I use MRI data, which stands for magnetic resonance imaging. Within MRI, there's different types of MRIs that you can do. So structural MRI is one of those, and it maps the structure or the morphology of the brain. These are used to qualitatively and quantitatively describe the shape, size, and integrity of gray and white matter structures. And it can be used to quantify properties such as total brain volume, surface area, gray matter volume, and cortical thickness. We can also use diffusion MRI to map white matter pathways in the brain. So diffusion of water molecules is constrained by obstacles such as macromolecules, fibers, and membranes, such that water diffuses parallel to these white matter tracks. So by measuring how water is diffusing, we can actually figure out where and how big these white matter tracks are. Using those track data, we can then actually derive what we call structural connectivity, which represents how connected two brain regions are through these white matter or neural fiber pathways. We also have what we call functional MRI, which measures brain activity by detecting changes in blood flow coupled with neuronal activation. So when the brain is consuming energy to produce some behavior or function, it is consuming a ton of energy, but it doesn't actually have a reservoir of energy. So it relies on what's known as neurovascular coupling and then hemodynamic response for oxygenated blood to be rapidly delivered to brain tissue. Measuring these patterns of activation, we can then look at correlations between local or regional neuronal activation patterns and use that to measure functional connectivity. So these functional connections represent statistical correlations in functional neural activity and tell us how similarly two brain regions are in their firing patterns. Using this functional MRI data, we can also identify what are known as functional networks. So as their name suggests, these are networks associated with different functions. So for example, a visual network, it includes different parts of the brain that include the occipital lobe and neighboring regions that are largely responsible for visual processing. Similarly, the somatosensory network surrounds areas around the pre and post-central gyri and tell us which regions are involved in somatomotor processing. And networks like this are known as unimodal sensory networks as they underlie sensory processing within a single modality. On the other hand, we have heteromodal association networks, which represent networks involved in higher order cognitive functions. So for example, the default mode network is the network that's largely active during rest or a default state. We also have things like the dorsal and ventral attention networks, which underlie different forms of attention. And in the neuroimaging analyses that we do, and specifically when it comes to functional MRI, we tend to describe our results in terms of these networks rather than specific parts of the brain or specific regions to try and simplify things. So I'll refer to these heteromodal association networks and sensory networks kind of throughout the talk. So going back to this idea of modeling, we can use patterns of functional network connectivity or functional brain connectivity in these descriptive, predictive, and prescriptive analyses. But when developing these models, there are a lot of different methodological choices that must be considered as they can directly and indirectly affect the model's performance and interpretations. Some of these choices include the neuroimaging modality and state that we choose to base our predictions on, the parcellations that we use, how we transform our data, how many subjects we include, whether we use population-specific or general models, and then the algorithms that we use, and finally, the phenotypes that we're seeking to predict. Today, I'll largely be focusing on analyses using resting state functional connectivity, and within that, I'll focus on how we can leverage population-specific predictive models to better understand relationships between sex, gender, and the expression of psychiatric illnesses. So this idea of population-specific modeling the basis is that when we're developing these machine learning models, we can choose to base them on the entire sample. So basically, we take all of the data that we have and use it to model these relationships. Or we can split the sample into specific groups based on some criteria like sex. Then we can actually model the relationships in each group or sex separately and compare how similar or different they are. And not only that, but we can then see if models trained in one sex generalize to the other one and vice versa. So it's a pretty straightforward and simple approach, but it's a great way to understand and quantify sex differences in brain behavior relationships, or the lack thereof them. And in terms of the behaviors that we're trying to predict, there are two main types of models used in psychiatry to represent different behavioral measures. Discrete models like the DSM provide us with diagnostic labels, whereas dimensional models describe broad features of illnesses across dimensions. And they provide complementary information, so using the two, we can get useful information that tells us not only about a diagnosis, but also about individual differences in symptoms. And this is important because there are a lot of sex differences that exist in the prevalence and the presentation of these illnesses. Based on the global burden of disease data, depressive anxiety and eating disorders are more common in females, whereas ADHD and autism spectrum disorders are more common in males. And these differences emerge at different points during the lifespan and tend to persist. But it's unclear whether these differences in the expression of psychiatric illnesses and the dimensional symptoms map onto sex differences in the brain. And in terms of these sex differences in the brain, throughout the lifespan, it's been well established that males have larger volume than females in terms of gray matter, white matter, subcortical gray matter, and ventricular volume. But the differences are not just restricted to structure. There are also differences in brain function. So this study has shown that females are more likely to have stronger within module or within network connections, whereas males are likely to have stronger between module connections or between network connections. If we look at the functional organization or the functional topography of the brain, this also differs between males and females and can actually be used to correctly classify males and females with an accuracy of almost 83%. And the most important networks for these classifications are actually the heteromodal association networks that I mentioned earlier, which are involved in higher order processing. Those same networks, the heteromodal association ones that are different across the sexes, are the same ones that are implicated in a wide range of psychiatric illnesses. So in terms of hypoconnectivity and hyperconnectivity between these different networks, they're implicated in anxiety, autism, bipolar, depressive, obsessive compulsive, and schizoaffective disorders, as well as ADHD and PTSD. But it doesn't just stop at psychiatric illnesses. Functional connections within these heteromodal association networks are actually also implicated in basically all behaviors across cognitive, personality, and mental health domains. So this tells us that the same part of the brain that is most consistently and reliably different across the sexes is also what's implicated in a lot of the behaviors that we care about. And those behaviors themselves tend to differ between males and females. So if we're looking at different things across motor, personality, or cognitive abilities, there are specific differences between males and females. But we don't know if these differences in the brain map on to differences in behavior. And going back to this idea that some aspect of our brain is involved through some mechanism to produce some behavior, we have to recognize that there are several different ways in which a sex difference may exist, and they're not all the same. So the differences may exist in the brain, in the mechanism or the relationship, in the behavior, in some combination of those factors, or not at all. So when studying specific behaviors, there might be differences in the brain that do not translate into differences in behavior. There might be just differences in the mechanism that translate to differences in behavior, but there's nothing actually in the brain. So in my case, I'm using machine learning predictive modeling approaches to understand how brain behavior and brain behavior relationships are shared or unique across the sexes. So today I'm going to talk about a few different studies where we've used these approaches, and I'll focus on aspects of cognition, mental health traits, and then end off with some ongoing work that I'm doing to look at gender. So starting off with cognition, I was interested in looking at how is functional connectivity or this functional MRI correlations associated with individual variability in cognition? And specifically, I was curious to see if this relationship between functional connectivity and cognition are the same across sexes. So this is earlier work that's been done from another lab, and they found that female IQ or female cognition was more predictable than male IQ based on overall functional connectivity. So on the x-axis, they have the true values of cognition. On the y-axis, they have the predicted values based on these modeling approaches. And they found that these predictions were more accurate in females than they were in males. They also looked at the specific correlates of cognition in terms of connectivity and found that males and females demonstrated unique associations between connectivity and cognition. So if you look at the circle plot on the right, in blue are the connections that were important in males, in red are the connections that were important in females, and in black are the ones that are shared. And there's actually only 3 connections that were shared. So they got to the conclusion that functional connectivity and cognition, the underlying relationship that mediates that behavior, is actually distinct based on the sex. So trying to see if that was the case, we basically repeated these analyses, but in a different data set and looked at different aspects of cognition. So we had functional connectivity data, we had aspects of fluid and crystallized cognition, as well as total cognition. And we built these models to basically take the entire data set, split them into males and females, and then see how accurately we could predict cognition. And we also actually did what we call sex-independent models. So this includes all of the data, as well as sex-specific models that were trained separately in males and females. And for all of these models, both sex-independent and sex-specific, we looked at how accurately those models could predict cognition in males and in females separately. So looking at the extent to which sex-independent models were able to predict composite cognitive scores, we first here along the x-axis, we have the different cognitive scores, and the first set is the total composite, and then we have the different crystallized measures, and then the fluid measures. And along the y-axis, we have prediction accuracy, which is a correlation between true and predicted values. And overall, we see that in terms of total and crystallized cognitive abilities, they are equally predictable for the most part across males and females. But fluid abilities are only successfully predicted in males, if at all. And this is actually the opposite of what the previous study found, because we're actually finding that cognition is more easily predicted in males rather than in females. So then diving in deeper, we did the same thing, but we separated the models in terms of the sex specificity. So we have models that were trained and tested in males in purple, trained in males, tested in females in blue, female trained male tested in green, and then trained and tested in females in orange. And here, again, we see that for total and crystallized abilities, the results are pretty comparable across the sexes. But these models generally fail to predict fluid abilities altogether, and when they do work, it's only in models that were trained and tested in males. So this kind of suggests that seeing these overlapping results across the sexes in total and crystallized, there might be similar correlates of behaviors correlates of cognition within those domains, but different across the fluid domain. So looking at how strongly those feature weights or those associations were correlated, we looked at all the different male trained models, all of the different female trained models, and correlated how strongly the relationships they captured were. And overall, we actually found that males and females had similar associations between connectivity and cognition, but there were slight differences in the strengths of those associations. So essentially, it's the same aspects of the brain, the same connections that are underlying behavior, but it could be that certain aspects of those relationships are stronger in one sex versus the other. So to try and summarize those studies, we see that crystallized and total cognition are comparably predicted across males and females, but fluid abilities can only be predicted in males if at all. But overall, it is these shared functional connections that predict individual cognitive abilities in males and females. And the reason why we wanted to do this is because in a lot of different clinical contexts, not just in psychiatric illnesses, but a lot of different illnesses where we do see cognitive decline, these models could potentially have utility in seeing how cognitive decline may occur. But if these models are trained on everyone, we may not be able to capture how accurately that's going to happen or how reliably it'll change over time. So this gives us an added layer of information that can bring it a bit closer to what precision medicine is actually supposed to be. So then moving on from cognition, we basically wanted to do the same thing, but then also look at predictions across these psychiatric illness-linked behaviors. So specifically, we first wanted to see whether functional connectivity patterns could predict the expression of behaviors linked to psychiatric illnesses in males and females. And here we specifically focused our analyses in children because we wanted to evaluate if these potential differences in brain behavior relationships emerge prior to the onset of puberty, as well as the associated changes that occur in psychiatric illnesses. We then also wanted to see if the specific brain behavior relationships we could identify were shared or unique across the sexes in terms of those network correlates and also what those network correlates were. And one additional thing that we did here was not only did we look at how these models generalized across the sexes, we actually also looked at how models generalized across behaviors. So for example, can a model that's trained to predict anxiety also predict depression and vice versa? But adding on the layer of sex, can a model that's trained to predict anxiety in females, for example, go on to predict aggression in males? Because we wanted to see how accurately these models not only capture the relationships, but if it's shared correlates across different behaviors modulating these relationships. So in terms of the data, we were using an adolescent data set. And in terms of the behavior, we had measures of internalizing behaviors, externalizing behaviors, thought, attention, and social problems, as well as total problems. And these kind of captured what I referred to as the dimensional symptoms earlier. And we also had measures from DSM-oriented scales, which captured things along affective, anxiety, somatic, oppositional defiant, conduct, and ADHD disorders. And part of the reason why we wanted to do both of these is because one, I mentioned they offer complementary information, but two is also that we might not see differences in just say affective or anxiety, but we might see things within these dimensional scales that we can't pick up otherwise. So here, once again, we had brain data, and using the brain data, we obtained resting state functional connectivity. And starting off by looking at the differences in behavioral scores, overall, we found that in both males and females, the behaviors are largely overlapping. So there were significant differences in a few of these measures, but overall, if you look at the distributions of all of those, there weren't any behaviors that only one sex or the other exhibited. They were both consistently exhibited. So this tells us that any results that we see are not just going to be because certain behaviors are only present in one sex and not the other. We then also looked at the correlations between these behaviors, and we found that males and females generally exhibit similar behavioral patterns. So this means that if you look at the correlations across all of these behaviors, there are obviously some that are highly correlated. So for example, rule-breaking and aggressive behaviors, but a lot of behaviors for the most part are not actually that strongly correlated. So this now tells us that if our behavioral predictions generalize across sexes or across behaviors specifically, it's not just going to be because all of these behaviors are strongly correlated. So getting into the modeling part, we used a similar approach as earlier, where we had sex-specific models to predict the 17 different behavioral scores across both sexes. And then we evaluated all of those models, again, across behaviors and across sexes. So getting into our results, we see that these predictive models do accurately predict psychiatric illness-linked behaviors. So on the left, we have results in males. On the right, we have results in females. And the stars here denote that they perform better than chance. And across all of the behaviors in males, we see that things within the externalizing domain, so externalizing, rule breaking, and aggression, perform better than chance, as well as attention, social, and total problems, and oppositional defiant conduct and ADHD. But we generally struggle to predict internalizing traits. In females, it's generally similar, but we are also able to predict withdrawn depressed traits above chance levels, whereas we're not able to predict total problems or oppositional defiant scores. So this suggests that there are some sex differences in terms of what behaviors we can predict and possibly in how those behaviors generalize. So then we looked at how do these models actually generalize and what does that tell us about the brain correlates of these behaviors. So there's a lot going on here, but I'm going to briefly define what we're showing and then talk about the results specifically. So model generalizability is what we measure when we're looking at how well a model performs when it's trained in a specific behavior or sex and then tested to predict something completely different. And all of the results that I'm showing here, everything that's shown in a color means that it performed better than chance across multiple comparisons across all of these different combinations, which was over 1,100 different combinations. So looking at these results, we have results from models that were trained in males at the top, trained in females at the bottom, tested in males on the left, and then tested in females on the right. So specifically looking at these models that were trained in males and tested in males, overall, we see that models that were trained to predict different externalizing behaviors. Just trying to see if I can find my cursor. I don't think it'll work. But models that were trained to predict different externalizing behaviors, like overall externalizing, rule breaking, or aggression, generalized to predict one another, as well as things like thought behavior or thought problems, attention problems, and social problems. These models then also could generalize to predict things like oppositional defiant, conduct, and ADHD. And this makes sense because a lot of these things are inherently correlated, and usually if you express issues across one domain, you might also be expressing it across another domain. Where it gets really interesting is when you now try to generalize these models to females. So here you'll notice that for the large part, those dotted boxes are not colored in. So what this tells us is externalizing behaviors, for example, cannot generalize to predict internalizing behaviors. But if you now test these models in females, we actually see that models trained in males to predict different aspects of externalizing behaviors do generalize to predict internalizing behaviors. So this now tells us that the same parts of the brain may actually be underlying these externalizing behaviors in males and internalizing in females. We could then validate that by doing the same thing but training the models in females. So if you look at these models that were trained and tested in females, we see a lot of the same trends, but overall these predictions are less accurate to begin with. But we do see that the externalizing ones generalize to predict other related ones, as well as things like conduct and ADHD. But these models don't really generalize to predict externalizing behaviors within females. But once we test these models in males, we now again see that models trained in females to predict something like a withdrawn depressed trait or overall affective problems, those models now also actually generalize to predict externalizing problems within males. So again, this tells us that the same parts of the brain that are underlying internalizing behaviors in females may actually be underlying externalizing behaviors in males. So then we went on to look at the specific neural correlates to predict these different behaviors. And overall, this was just, again, correlating the different feature weights from the models. So it tells us, if you look at across all of these models, they are largely correlated. So essentially, there is a lot of overlap in terms of the brain networks that are underlying these different behaviors, both across males and females. So there's a lot happening here. So we're going to actually talk about what those correlates are. And to try and simplify it, I'm going to be using the rule-breaking one to represent the externalizing behaviors and the withdrawn depressed one to represent internalizing behaviors. So here we have the associations between functional network connectivity and these behaviors. And on the top, we have positive associations, which means greater connectivity means greater problems. And on the bottom, we have negative associations. And on the left are results for males, and on the right are results for females. And overall, we see that both within the positive associations and the negative associations, the patterns are largely shared across the sexes. And for the most part, we're again seeing these connections within and between different heteromodal association networks, such as temporal parietal, default, control, limbic, and attention. So basically, all of these connections are the ones implicated in rule-breaking behaviors. If we now shift to look at the withdrawn depressed behaviors, these correlates you'll see are largely the same if I just go back and forth. So this now again tells us that there must be shared neural correlates that underlie these behaviors, so these internalizing behaviors, as well as externalizing behaviors across both sexes. And perhaps how they are manifested is driven by other factors. So to summarize that, in children, functional connectivity predicts externalizing behaviors in males and females, but only predicts internalizing in females. And these models that are trained to predict behaviors within a given domain generalize to predict related behaviors in the same domain, both within and between sexes. But they only generalize across domains across sexes. And across all of these different behaviors, it's these functional connections within these heteromodal association networks that are underlying their expression. So based on all of this, we think that males and females may actually be defaulting to gender-specific socially acceptable presentations of the same underlying mental distress along a spectrum of internalizing and externalizing. But that then leads me to point out that sex is not the same thing as gender. So in the work that I've been doing, I've typically considered sex assigned at birth, which is based on an individual's physical anatomy, physiology, genetics, or hormones. On the other hand, gender is a multidimensional construct that relates to the attitudes, feelings, and behaviors associated with a particular sex. And both our brain and our behavior are shaped by both sex and gender. So in the work that I'm doing right now, we're actually trying to disentangle the brain correlates of sex and gender. So I'm going to briefly talk about that, and then I'll wrap up so we have time for questions. So like I said, we wanted to see if functional connectivity can predict sex and gender, and we wanted to see what are those brain functional correlates of sex and gender, and are they the same or are they different? And here, once again, we were using the same data set as earlier, so these are results from children between the ages of 9 and 10. So looking at the distribution of gender identities, we see that both in terms of self-reported and parent-reported gender identities, males reported stronger sex-congruent gender identities than females. So what this means is males tend to identify more strongly in line with their sex in terms of their gender identity and expression than females do. And these results are in line with prior work that's been published that, especially in kids, there is a greater pressure to exhibit sex-congruent gender identities than there is in females. So looking at if functional connectivity could predict sex and gender, we again train these models across all of the individuals to predict sex and to predict gender. So here I'm showing the results both in terms of how much of the variance does functional connectivity explain in sex and gender includes separately the measures for self-reported data as well as parent-reported data, as well as the prediction accuracy. And unsurprisingly, functional connectivity captures a lot of the variance across sexes, and our prediction accuracy is around 75%. So this is nothing new and very much in line with what's been shown before. In terms of gender, we see the same results, and what I've done here to predict gender is I actually took these measures and the female scores were reversed. So the scores went from 0 to 20, 0 indicating a more feminine gender expression, 20 indicating more masculine for the self-report, same thing for the parent report, but it ranged from 0 to 60. So looking at this, you'll probably say that these predictions of gender, if you're doing it across everyone, are probably biased because we can't be sure if it's capturing sex or gender. And that is 100% true because sex and gender in this data set are very highly correlated. So instead, we wanted to predict gender in a sex-specific manner to then actually capture the sex-specific correlates of gender. And here we see that, again, functional connectivity can successfully predict gender, but we're only able to successfully explain the variance and predict it accurately in terms of the parent report data and not the self-report. Again, earlier I showed you these figures of these distributions. The two are very different in terms of how they're distributed. The two are also very different in terms of the types of behaviors they capture. So that might perhaps be explaining why our results in both females shown at the top and males shown at the bottom are only accurate in parent report and not the self-report data. But we're not sure if that's what's driving it or if there's something else going on. So then looking at the functional correlates of sex and gender, we again looked at the correlation between those brain correlates across all of the different measures. So we have the results from the sex prediction, the results from the self-report and parent report across all individuals, and then the sex-specific results from the self-report and the sex-specific results from the parent report data. And overall, the sex and the gender predictions across all individuals are strongly correlated, which makes sense because we think that those predictions of gender are actually just relying on this underlying sex information. But if you now look at the self-report female, self-report male, parent report female, or parent report male predictions, those are very different from those observed with sex. So this now tells us that there must be distinct functional networks that are associated with sex than those that are associated with gender. So we looked at what those networks actually were. And visualizing those correlates, here the heat map shows the absolute associations between connectivity and sex. So the warmer colors indicate stronger associations and cooler colors indicate weaker associations. So basically, warmer colors are more important to identify an individual's sex. And overall, we see that sex is associated with functional connections within and between both unimodal and heteromodal associations. So we see combination or contributions of control, limbic, somatomotor, and visual networks. Then, if we look at the correlates of gender, we see that they're pretty different. So here, these ones are more strongly emphasizing these heteromodal association networks, and this is in both females and males, and across the sexes, there are some slight differences in terms of these associations, particularly that in males, we do see a stronger contribution of these visual and somatomotor or sensory networks than we do in females. But overall, these networks that represent the underlying variability in gender identity and expression are very different from the networks that are implicated in our sex. So to summarize, this tells us that functional connectivity can predict both sex and gender, but there are these distinct functional correlates associated with sex and gender. And the functional connections that are implicated in sex are those within unimodal and heteromodal networks, whereas those important for gender are primarily located within these heteromodal association networks. And once again, to try and bring it back to what I presented earlier, what's really interesting is that these same correlates that distinguish individuals based more so on gender than on sex are the ones that are implicated in different behaviors such as cognition, as well as the ones linked with psychiatric illnesses. So if we're trying to understand these brain behavior relationships, and especially if we're trying to understand these relationships that involve these networks that are so different across sexes and genders, we need to take into account sex and gender before we can actually develop a better understanding of the brain correlates of those behaviors. So to try and summarize everything, these brain-based predictive models facilitate the study of brain correlates of behavior across both healthy and clinical populations. And these population-specific and sex-specific models allow us to study how shared or unique brain correlates underlie behaviors across populations, such as across sexes. And these heteromodal association networks differ across sexes and are also implicated in a wide range of behaviors, including cognition, internalizing and externalizing behaviors, as well as gender identity and expression. So these heteromodal association networks may be a potential biomarker for personalized diagnostic tools and preventative strategies for these illnesses. But why does all of this matter in the context of women's health? Well, if we now start to consider sex and gender both independently and interdependently, this will shed light onto how these variables influence not just our behaviors, but also our brain development and organization and that relationship between brain and behavior. And if we start to get an understanding of these female-specific brain behavior relationships, we can then start to consider how other factors that are sex-specific, like sex hormones, can influence these relationships. So, establishing these female-specific biomarkers of psychiatric illness risk and related behaviors will then be critical for the improved prevention, diagnosis, and treatment of these illnesses, which often exhibit unique patterns of expression in women that we don't see in men. And with that, I want to thank all of my collaborators and my funding sources. And for anyone out there that might be looking for a postdoc, I am currently recruiting people to join my lab. So if you're interested, definitely reach out. And thanks again for the opportunity to present this work.
Well, yay. Thank you so much. I know it's so hard virtually, so if you can give a little emoji clap, I would appreciate it. And I do see some already. Oh, I see many more now. So thank you very much. Everybody's looking for their little emoji. So yes, you're getting a round of applause now, a virtual round of applause. I meant to mention earlier that I usually tweet or X or post or whatever we're supposed to call it, but I won't. I didn't post anything that was still in prep. I have a couple of questions. It's more about not necessarily questions, but thinking stuff for us. But I also urge any of you, if you have any questions, to either put it in the chat or put your hand up. And because it shouldn't all be about my questions, especially since I am not a machine machine learning modeler. But every time I listen to Elvisha, I think my brain grows a little bit more. So I'm hoping by the fourth time or so, it will grow even exponentially larger. I have some questions for you, and some of them I think are going to be a little bit unfair, because it's not a data set that you can really move too much, right? So, or you can change. So it's all based on the questions that were asked of the individual, right? So I'm thinking about what you said, something about like the gender. I didn't want to go this first, but the parents' gender assessment versus the child's self-report, and that they're different. I think that's really interesting, and what those questions are and how they might be different, or would be really interesting to sort of dissect out. But that's, I know, not your specific area of what you really want to look into. What I was thinking when you were talking about the externalizing and internalizing, I was wondering how because I noticed, I think if this is true, so I'm talking about the first part of your work first, when it's trained and tested in males, they were more accurate. Is that correct?
Yes, they were.
So then I started thinking, is it because of the questions we're asking, right? Like, so if our questions have always been based on what's going on in males, maybe that's why it's more accurate, and we need different kinds of questions for or we just need different kinds of questions. It shouldn't be about males and females, but it just if our sort of work has been, and I don't mean your work and my work, I just mean everybody's work has been focused on what's going on in males, maybe we need some further questions. And even what we call internalizing or withdrawn depressive, what does that mean? So this data, is it based on what data set?
So this is all ABCD, so adolescent brain cognitive development. And something that is related to what you mentioned is also, in general, the predictions, not just for males, but specifically for externalizing behaviors, are more accurate. So, and I think it's also because, especially within that age range, it's easier to kind of measure and understand what these externalizing behaviors are, as opposed to trying to explain to a 9 or 10-year-old what like anxiety is and depression is. And it's going to mean very different things, especially in populations where it's kind of the same thing about how like ADHD tends to be diagnosed far more in males than in females, because males exhibit the more externalizing aspects of that diagnosis, whereas females have the more like internalizing subtype of it. So I think it's not just within this issue of like the sexes, but also the behaviors and the measures. And we're actually going to be repeating all of these analyses at the different age groups. So there's longitudinal data collected in these kids every other year for 10 years. So about now, these kids are 13 to 14 years old. So as these data get released, we do want to see, does this trend also hold throughout development when, as kids get a little bit older, go through puberty, have a lot of these changes, they might also express things within that internalizing more so that makes it potentially easier to predict.
Oh, that's so interesting. Yeah, 100%. Like, I think as time goes on, it'll be interesting to see whether the model holds up or if it changes, right? And it could be changed based on age. It could just be changed based on exactly what you're saying, that it's the measure itself is more accurate, right? The measure
Absolutely.
of the behavior, right, isn't more accurate because they're a little bit older. So that's what I just assumed, that this is in adults, but of course it was, if I remember it, I would have thought about it a little bit more. So these are almost all pre-puberty then, all of the data?
Basically, yeah. So they're all 9 to 10. So some of them may have started puberty. We haven't explicitly looked at any of the puberty measures. They have collected some kind of like saliva measures of hormones as well as self-reported Tanner staging. But it's kind of like a single snapshot rather than a detailed overview. So it's also hard to then get a decent enough understanding of how those hormones might be influencing it, because it's not necessarily measured in the best way either.
No. Yeah, exactly. No, but I'm wondering whether or not you have an interest, or you would predict, because you're such a predictive modeler, whether you'd see these same kinds of relationships later on in life. So could you do this? Like, this is, a really interesting tool to do earlier in life, but I'm wondering later on in life if the same or maybe they'd be even stronger, the either the predictive accuracy or the fact that you see male trained data on male tested, whether those kinds of relationships would differ across time and age.
Definitely. I mean, we definitely kind of going into the like longitudinal aspect of this that we're going to be working on. We do think that as they get older, the internalizing aspects will be easier to predict.
Right.
And then we're also then trying to figure out here, obviously, everything is just based on their sex assigned at birth, but then kind of expanding that to then also consider gender identity, because we think that as the individual variability in gender identity also develops, there will also be specific aspects of these behaviors that might be seen more in people that identify with a specific gender that's more representative of who they are than just their sex assigned at birth.
Mm-hmm. Absolutely.
So kind of trying to bring all of those ideas together.
So what are the, since we're talking about this now, do you know what some of the differences are in terms of the gender scale? Like you said, I think it said it was 0 to 20. I wrote down a lot. These are my there's a lot of questions on my computer. Some of them I can't quite read, and something about the card sort, but yeah, 0 to 20 for self-report.
So the self-report is 4 questions. Each question goes from basically 0 to 5. And the questions are kind of like, I don't remember the exact wording, but it's like, I identify as a female, like all the time, most of the time, some of the time, and so on. And then I identify as a male, same thing. I think one of them is like, I've had the desire to be a male if you were a female, or vice versa. Whereas the parent report, and the self-report, I should mention, is what they call primarily gender. They call all of these gender identity scores, but the self-report is what's actually capturing gender identity, whereas a parent report is what's kind of been referred to as gender expression, gender dysphoria, and it captures things like and since it's parent reported, it's like my child often plays with girl themed toys like Barbies, or like male, like boy themed toys like G.I. Joe. There's also things about when my child is playing house, they often will take on like the girl role or the boy role. There's things about how they dress, how much they're interested in things like makeup. And the questions themselves obviously are not necessarily the most accurate
I was gonna
representation.
say,
Then,
I know, yeah, some of the gender role, and so apologies to those of you that might be in the gender field, but I think this is something that we all need to think about when especially if we're researching gender, like what are those questions and what do they really mean? Like, does wearing an apron when you cook really mean anything? Or horseback riding,
exactly.
another one I heard about recently, and I was like, what does that have to do with anything? And the fact that
And then it's also, you add on the additional bias of what the parent thinks is stereotypically male or female.
Right. And even playing with a G.I. Joe. Like I had my Kens playing with Barbies. I mean, I mostly played with the Barbie, but sometimes the Kens played with they were, I don't know if you've seen the Barbie movie, but they were slightly less interesting than the Barbies. Let's say, had fewer outfits to put on. Yes. So that, I think it's really, really interesting. And you're limited by, we're all limited by the ways that we're measuring gender.
Yes.
But what about things like, would you be able to look in the ABC data in terms of, rural versus urban areas and how that might differ?
We do. So we're currently working on this paper. We do also have the same figure, but separated by site. Okay. So obviously there's like 20 something sites. They're in very different locations. But then you also get to the issue of the sites don't necessarily represent the people that live in those areas.
Right.
Oftentimes the people that are recruited are like someone that works at the university and their kids.
Right.
We don't want to draw any conclusions on site at this point. But we are also interested to, again, look at these data kind of throughout the course of development. So something that I forgot to mention actually is these gender data were not collected at the same time as the brain data. They actually added it on a year later.
Right. So
they're then also going to have to change these questions because when you're 16, you're probably not going to ask if your child is playing with Barbie. Right. So it'll also be interesting to see how those questions change and what we decide is something that is representative of gender, and then also how these distributions shift over time. Because it's in line with the changing world because these kids are growing up now and we're talking about these things more.
Right. Well, yes. The other thing that I think, I have a lot, I just don't want because I know we don't have that much time because there's only 2 more minutes, but the other thing I notice about this particular scale is, they're almost all of them in the self-report are on the 20 side, right? So that makes me think, are the questions we're asking, I'm not saying anything other than it seems like the 20 part of the scale is more exciting than anything else. So that makes me think that maybe it's not a very good capture, and maybe they're just too young a capture of gender.
Because it's like, I think part of it is definitely, I don't know to what extent these kids are explain these questionnaires, but also they have to fill out so many questionnaires that if I was them, I would probably also just choose one number and go all the way down.
Yeah, no, there's also a part of that. Absolutely. Well, I see that we have 1 minute left. So I just want to reiterate how fantastic was. I wrote down a lot. I still have to tweet a few little things about some of the things that you said, or post, excuse me. But we really appreciate your work, and we really appreciate you taking the time to talk about your work with us today. It's really fascinating. I always just see I always have a lot of hope in the future when I see this kind of work, especially when people like you are paying so much attention to doing your predictive modeling and training it on males and testing it on males, testing it on females, and looking at the differences there. And so thank you so much for the work you do, and looking forward to hearing more about it in the future.
Of course. Thank you so much for the invitation.